We heard already really great science from Kaiji and Mike, and obviously there are many people in the room that have a lot of experience with cardiomyocyte implantation, and one of the key lessons we have learned really over the past 20 plus years is that if you want to put muscle into the heart, you do have to implant cardiomyocytes. This was done in the mouse very early by Lauren Field in this group, and then obviously excellent studies with human ES and IPS cardiomyocytes in the monkey from Chuck and Mike from his time when he was still in Seattle, and later Mike's work in the pig were really fascinating and instrumental to demonstrate that that the heart can be remuscularized if you put in the right cell. The challenge, of course, and this was also discussed already, is how to do that safely and with maximum efficacy. How can you have a large number of cells in the heart without inducing arrhythmia, which is a key problem, of course, in the field. And we heard about a number of interesting approaches already: maturing, genetically modification, which apparently did not work so much, but maybe maturity is a point, because we are not working on cardiomyocyte implantation. We are working on implantation of engineered heart muscle, which we believe is a way to mature cardiomyocytes quite nicely. And in addition, we are not focusing on implanting these cells directly into the myocardium, but onto the myocardium to add muscle from the epicardial side to the heart. And one of the key lessons we learned through this process is that it is, at least in our hands, we would not go for optimal purity to achieve maximum maturation. We think that it is important to have at least a stromal cell component added to the cardiomyocytes. We like to add fibroblasts or fibroblast-like cells, which are able to modify stroma, which are able to condense the matrix environment, and by doing so are able to put mechanical strains and stresses on cardiomyocytes to support their maturation. And this is demonstrated in this experiment, and we have done this actually in many species, in mouse and in rat, and monkey and human. It's always the same. So if you use very highly purified cardiomyocytes, you will have cardiomyocytes located inside, in this case, a collagen matrix. These cells will also beat, but they will not form a network. So you will have a lot of independently beating cardiomyocytes, but not a network. This is what you see on the right-hand side. And these cultures were done in parallel. And the only difference is that in this culture, we added human foreskin fibroblasts. I should also add that it doesn't, in our hands at least, it doesn't really matter if you put in cardiac fibroblasts, human foreskin fibroblasts, skin fibroblasts, whatever fibroblast, as long as the fibroblasts are capable of modifying the matrix environment, then they basically do the job on the cardiomyocytes. It's probably very much a mechanical stress which is helping to mature the cells. And with all that information gained over the years, we basically identified ideal cell compositions. I'm afraid the pointer doesn't work. We identified ideal cell compositions which allowed us to form engineered human myocardium with optimal contractility. And that is, in our hands, always the case if we have roughly 50% cardiomyocytes and 50% stromal cells in the end in the tissue. When we construct our tissues, we are not putting in 50-50, we're putting in 70-30, 90-10, depends a little bit on the format. And there is a change in cell composition, and it always ends up at 50-50, at least in the models that we are using. And if we do that, we achieve tissues which do develop quite nice contractility, and not only that they contract, they also mature functionally over time. What you see on the right is an EHM culture for 2, for 4, and for 8 weeks, and this is force of contraction. under increasing calcium concentration, and you can see that the force of contraction is increasing under increasing calcium concentration, and then the older the cultures are, the more force they develop. And if you compare now the forces in the EHM to the forces in myocardium, which was taken from infant heart muscle or newborn heart muscle in adult, you can appreciate that the forces the EHM develop are pretty much in between the forces that you would see in postnatal human myocardium and adult myocardium. And this I'm not showing, but if you look into cell dimensions, expression patterns, and so on, we believe that we interpret the findings that the tissues we are forming represent roughly the myocardium of a 9 to 12-year-old. So it's young muscle, but if you look into these structures and compare it to published data, it's clearly postnatal in many aspects. And by now using this knowledge, cell composition, and using collagen, defined collagen, and the casting procedure, we can basically create tissues of pretty much any size. This is a smaller ring format, which we typically use for drug screening or disease modeling applications. On the right-hand side, you can see tissue formats that we are presently using as the minimal dose unit in our clinical applications. And in addition to just being able to make tissues of any size in X and Y, these tissues can also be stacked and can basically be stacked to also pretty much any size. That sounds a bit strange, but it is not, because we have the way how we design these tissues is that the openings and pores you see here provide enough exchange of medium to not create core necrosis, even if you would make a tissue of 1 meter thickness. So this is a simple calculation basically that you can do, and by choosing the appropriate porosity, you can basically create anything in X, Y, and Z. And moving this into a clinical application, we were challenged by our regulatory authorities that asked us to not consider implantation of human cardiomyocytes in large animals. There's a lot of different opinions about that, and this is all fine. But at least in Germany, it was very clear that they asked us to simulate a clinical trial as good as possible in a large animal model. And that was in 2014, and by that time we already had the pleasure to work together with our colleagues here at the Primate Center. We knew that we were able to generate iPSCs from rhesus macaque, and what you see here is a human-sized macaque patch, and this is in scale. This is the monkey heart. This is the patch heart, the patch. And you can appreciate that we were able to also construct rhesus macaque EHM for implantation studies then in the monkey. And to do that, to identify appropriate dosing and how this should be simulated well, you have to consider the differences in size of the heart. So the monkey heart is roughly 10% of the weight. This is the left ventricular weight of the human heart, and the target heart wall thickness is roughly 50% of the human heart. And with that, we developed or designed a preclinical study first in healthy monkeys, where we tested whether or not the implantation of EHM could be done safely, and whether or not the implantation would lead to an enhanced an augmentation of the target heart wall. You can see a patch here, which is on the surgeon's hand, which is then moved onto the heart through a type of mid-cap left lateral thoracotomy. It's moved onto the beating heart, and this is 1 month later. This is 3 months later. This is a small dose, and on the right-hand side you can see the target heart wall thickness. This is investigated by MRI, and you can appreciate that the target heart wall is thickened by roughly 1 millimeter per layer of a patch. So one patch thickens the heart by 1 millimeter, 5 patches roughly by 5 millimeters. So that was encouraging. There were no arrhythmias, no side effects. Tested this in 14 monkeys, and that went all quite smoothly. And then, of course, the question came up whether or not. Don't worry, I'm post-COVID. I still have a bit of a post-COVID cough. We then simulated the study in a heart failure model, which we had to set up first. That was not existing, at least not a chronic heart failure model. And we were taught by Chuck and his colleagues how to basically do the infarction in the monkey, by Chuck Murray, and do the monkey model. But then we did not implant after a week or two. We waited for 6 months. We waited for 6 months to see whether or not a heart failure phenotype would develop. And then, if it would, we then randomized the animals into 4 groups, into 2 control groups, 1 without and 1 with immune suppression, and 2 groups with a mid dose and a large dose of implant. And you can see that here, this is the implantation procedure. Here again, the picture of a patch moved onto the heart. And this is summarizing the findings. On the left-hand side, pathology of the heart. You can see one of these patches here, located nicely at the anterolateral side of the left free heart wall. In the cross section, you can see that there are quite a few cardiomyocytes deposited on the heart. This is a Desmond stain in brown. You can see the damaged area here, and you can see the implanted area by these encircled patches. So roughly 70% of the cells that we implant could be found again after implantation, which is quite nice, and may also be due to maturity, as we have heard it earlier. What happened then with the heart MRI data? Again, target heart wall was thickened, also in a dose-dependent manner. These are now 2x formulations and 5x EHM formulations, a pretty similar observation as in the healthy monkeys. And most importantly, this is now the anterior wall thickening fraction, the contractility of the target heart wall. This is what happens after infarction. That goes dramatically down. You have basically a scar forming. The target heart wall is not contracting anymore. And then the patch is implanted. And it takes roughly 1 to 2 months in this high dose group to reintroduce contractility. That was done in 3 animals. In 2 animals, we implanted the patch strictly on the anterolateral wall. And in the 3rd animal, we implanted the patch on the apical side of the heart, on the bottom of the heart. And as you can see here, and this didn't do anything. So it seems pretty important also to put in the patches in the right location to be able to support the contractility of the heart. And these 2 animals also had a better ejection fraction. You can appreciate that this is not an instantaneous process. This is a process that takes some time. So 1 to 2 months to actually nearly 3 months to see that the contractility of the patch is then supporting also global heart function. And that does not happen if the patch is in the wrong position. This is a summary of those 2 animals. Again, low N number, but it's a longitudinal study in which we have done MRIs all at many time points, and we are quite confident that this is a clear enhancement in those animals. So this is the monkey data I wanted to share with you. We have good evidence for muscularization. We see that the target heart wall is not only thickened, but that there's also contractility after implantation. We can see that global heart function is improved. And this all seems to matter also as to where to put the patch. We have done a number of numerical simulations together with our colleagues at the Max Planck for Dynamics and Self-Organization with Ebert Bodenschatz, which are in support of this finding also that the location does matter. I didn't have time to show you vascularization. There's good vascularization by evidence by histology and gadolinium perfusion MRI, and there's also good evidence for a reversal of dilation. The surgical procedure is through a pretty standard mid-cap access route. It is invasive, but it's considered in surgical terms a minimal invasive procedure. We didn't see arrhythmias, we didn't see tumor, and we didn't see a compromise of heart function. So with that data and a lot of data also on production and quality, we have a clinical trial approved, the BioVet-HF trial, which has started in 2021 at the midst of our COVID pandemic. This was also a pretty big challenge. But it has started, and it's an open label, multi-center combined phase 1-2 trial. The plan is to implant 43 to 53 patients. That depends a bit on the dose finding cohort, on the speed we can go through the dose finding. We were considering 8 to 18 patients, and after that we would go on an extension phase 2A type of study. And the dose levels are 200 to 800 million iPS-derived cardiomyocytes and stromal cells in a PET format. And these patients are on study for 12 months, and then they go into a registry until end of life. First patients have completed the 12 months already. They are now in the registry. We are now at the highest dose level at 800,000 cell units or cells implanted through the patches. And our belief that we would probably be able to finish the dose finding cohort with 8 to 9 patients instead of the originally planned 18, and the centers that are participating are Göttingen, Lübeck, and Bad Oeynhausen. And this is just a small glimpse into the operating room. These are the dose units. We have completed the low and the mid dose and are now studying the high dose applications. And just to give you a little idea how that looks like, this is how these patches are brought into the operating room. Actually, we had to design all these things ourselves because there was no appropriate primary packaging material available. So all the stuff that you see here is home built and approved by regulators. This is the container for shipping. Then you can see here the patch being moved out of this container. You can see here already a number of patches ready for implantation, and these patches are individually demounted from these stretchers and then sutured together in this format on a collagen membrane, which is then implanted on the heart. And you can see that there's already one implant located on the heart, and the second one is located just next to it. And this is covering 100 square centimeters of the heart and 800 million cardiomyocytes at the moment. And of course, super exciting. Sometimes also a bit scary, but a very rewarding study. And we hope that by doing so, we can position a great number of cardiomyocytes on the heart to support heart function. We are not dogmatic as to how to do it. I mean, there are certainly other approaches that will also remuscularize the heart, but this is one approach that we are pushing forward. And in the end, you never know whether or not it's a simple type of patch or a combination of cells and patches, a combination of paracrine whatsoever. I think there's a lot of opportunity in this space, and this is also why we're here and we are discussing these things. Again, the team is most important. I would like to point out Malte in particular, because he was very instrumental in transferring the lab process into the GMP environment together with our colleagues from Reparon. And obviously there's a large number of colleagues involved in this study throughout the whole clinic, also at the Primate Center, and also a number of national and international collaborators with whom we work very closely together, and will also advance this trial to an international multi-regional trial to then go beyond the German borders. Thank you very much for your attention.